guys. This little video clip is going to be an Exam 3 review. I hope all of you are well and healthy. It's been a little bit since we've gathered together and talked about micro. So today we're going to do an exam review. This covers chapters 16, 17, 18, and 19. So I'm gonna go over these kind of thorough because it's been a while since we talked about them. I want us all to be on the same page. You will be taking exams in the near future again, and I want you all to be ready. So let's start with Chapter 16. Chapter 16 is the group of organisms that are aerobic gram-positive bacilli. There were two main categories. There's the non-spore forming category and then the spore forming category. And under the non-spore forming category there were three subgroups. The three subgroups are non-branching catalase positive, non-branching catalase negative, and then branching. And then under the spore forming group there is non-branching catalase positive, and that included the Bacillus species. So let's talk about the first category, group 1. So non-spore forming, non-branching, catalase positive. There are three groups or different species types that are in this non-branching group. The first one is Corynebacteria or Corynebacterium. The second one was Rhodococcus, and then the third one was Listeria. So let's talk about Corynebacterium. These are, there are quite a few of these organisms. They are, of course, gram-positive bacilli, non-branching, catalase positive. There are three groups that are closely related, and they're known as the CMN group. And this includes Corynebacteria, Mycobacteria, and Nocardiae. So remember about that CMN group and which three big groups are included in that. So let's talk about the primary or premier pathogen of this group. But before I get into that, let's talk about what it looks like under the microscope. In a gram stain, these are palisading Chinese letters that look like L's or V's. They can look like a club shape, or they're also referred to as diphtheroids. So just remember that these are our palisade Chinese letters. They grow on blood agar plate, they'll grow on a Cy slant and Loeffler medium. There's also some selective and differential mediums for especially our premier pathogen, which is Corynebacterium diphtheriae, and that is Tinsdale agar. Colonies will appear blackish brown on this agar. We also have something called cysteine tellurite blood agar, which is another selective and differential agar for Corynebacterium diphtheriae. So there are two forms of diphtheria disease caused by Corynebacterium diphtheriae. The first one is respiratory. This one has a well-known trait that's called a pseudomembrane. So there's a toxin produced by diphtheria species. Not all produce the toxin, but the toxin-producing species cause diphtheria disease. And so this toxin causes tissue necrosis in the back of your throat area, nasopharyngeal area, and it results in this thick, tough, grayish pseudomembrane. The second form of diphtheria disease is cutaneous, which is more prevalent in the tropical areas. So another important note is that these organisms under the gram stain may appear beaded when stained. And when you stain them with methylene blue, it will stain their metachromatic granules, These granules in the cytoplasm are also known as Babies-Ernst granules. So know that, be familiar with that. There is a test called the Ehrlich test. And if you can remember, this detects toxigenicity of Corynebacterium diphtheriae via an immunodiffusion paper. So what happens is when they create this particular agar that's used in the Ehrlich testing, they put this this paper strip that is infused with an antitoxin. They put it slightly under the surface of the agar as the agar is still liquid, and so it stays submerged as it hardens or it solidifies. So when you streak at a 90-degree angle a known toxin-producing strain, and you streak at a 90-degree angle a known non-toxin producing or toxin negative strain, along with your, which is your controls, you'll also streak at a 90-degree angle your unknown Corynebacterium. And after incubation, this unique testing will display a precipitin line at a 45-degree angle in between your immunodiffusion paper and your streak lines. You'll have this 45-degree angle line of precipitin, and this would be positive reaction indicating that your organism is producing toxin. It's agglutinating with that antitoxin. So it's a really unique test for Corynebacterium diphtheriae. That's the Ehrlich test. So be familiar with that. The most common cause, there are a few other species, Corynebacterium jaceum, Corynebacterium ulcerans, Corynebacterium pseudotuberculosis. Those are some others. In particular, Corynebacterium jaceum is the most common cause of diphtheroid prosthetic valve endocarditis in adults. So you have to remember the most common cause of everything. Let's go on. So that's enough about Corynebacterium. Let's talk about the second group of species, and this is the group of called Rhodococcus. There's one in particular that I want to discuss, and that's Rhodococcus equi. This organism has a distinctive salmon pink colony color. So know that. That's unique to this species. And then the next group that is a non-branching catalase positive is Listeria. There's one very important group that we'll talk about, and that's Listeria monocytogenes. This organism is beta hemolytic. It is very similar to Streptococcus agalactiae or group B strep, except you need to know that Listeria monocytogenes is catalase positive, whereas group B strep is catalase negative. So know that these are very similar looking on a blood agar plate, except for that catalase reaction. Listeria is a cold enriched organism, which means that it can grow over a wide temperature range from 2 to 45 degrees. So sometimes we can recover this organism with cold enrichment, so we can grow it at colder temperatures to get it to grow. This organism is important clinically because it causes and can cause listeriosis in pregnancy, usually the 3rd trimester of a pregnant lady, and or can cause neonatal listeriosis. Neonatal listeriosis, there's 2 forms. One is early onset, the other is late onset. No, there's a very important trait to this organism. It displays something called tumbling motility. Also, there is a test, the Listeria Camp test. that produces block-type hemolysis when you streak it on the plate with Rhodococcus equi or Staphylococcus aureus. So unlike that previous camp test we talked about with the arrowhead hemolysis pattern, Listeria also produces that enhanced hemolysis effect, but it's a block pattern that you see. So know about that, and that it's related to Listeria. So let's move on to the second category of non-spore forming, and that is the non-branching catalase negative. There's one main group in this one, or one main species in this one that we talk about, and that's Erysipelothryx ruseopathii. It is gram-positive bacilli, non-branching, catalase negative, primarily causes disease in animals. However, it is an occupational hazard if you work with animals. It can cause septicemia, endocarditis, something called erysipeloid, which is a skin infection. There is a distinguishing characteristic I want you to know and remember about this species. Erysipelothryx ruseopathii is H2S positive on a TSI slant. So it'll have a black butt and it is H2S positive. The next group of non-spore forming gram-positive bacilli is branching. And under our branching group we have a large phylum of gram-positives called Actinomycetes, and two particular groups that fall into that Actinomycetes phylum is one Nocardia species, and two a group of others that I have talked about. There are several different species in that one, but we'll get there. So in our Nocardia group, these are branching filamentous rods, gram-positive rods. They resemble a fungus. They weakly acid-fast stain. They have a beaded appearance on the gram stain, so know that. There are two primary infection types caused by Nocardia. There is a pulmonary infection type, and then there is a cutaneous infection type. The cutaneous infection is usually caused by a species called Nocardia brasiliensis. This particular cutaneous infection causes something called mycotomas, so know that term. They have a dry breadcrumb appearance on agar, and they have a unique plowed earth smell. So this is unique to them. They smell like plowed earth. There are another category under that that's called the others, and that includes Actinomydura species, Streptomyces species, Gordonia, and Sucamurella. So there are others in this big phylum of Actinomycetes, but Nocardia is one that you'll clinically see commonly. So our last group, which is our other main category under the aerobic gram-positive bacilli, is the spore formers, the spore forming group. So these are non-branching, catalase positive, and there's one main category group of species, and it's the bacillus. It's the bacillus family. There are two primary pathogens that we talk about, and that's Bacillus anthracis and Bacillus cereus. So let's get into the bacillus family. These are unique in that they form spores. They form something called endospores in aerobic conditions. Bacillus is a family of organisms that is ubiquitous in nature, common out in grasses, in the dirt, farmlands. They're all over. However, Bacillus is usually only causes issues in humans when we're exposed to animals, animal products, or their hides or their wool, animal products. So let's go back up to endospores. So Bacillus is unique that it produces spores. And we can group Bacillus based on the location of their spore. So the spores can be located terminally. So the three groups are group 1, 2, and 3. Group 1 is Bacillus species that have a centrally located spore, endospore. Group 2 can have central or terminally located spores, and group 3 just primarily has terminal endospores. So we group Bacillus based on their endospore location. They oftentimes look like bamboo rods under the gram stain. They're very blocky. On a agar plate, they will appear like a Medusa head colony and oftentimes have this beaten egg white appearance. So know that those are two unique characteristics to Bacillus. The first pathogen that we need to be concerned with is Bacillus anthracis. This organism is non-motile, unlike the other Bacillus species. So this is one way you can differentiate Bacillus anthracis from the others in this group. They're non-motile. And of course, they're non-hemolytic on blood agar plate. Bacillus anthracis causes the disease anthrax, which I'm sure you've heard. Anthrax is caused by a toxin, which is very potent. So the anthrax toxin has 3 parts that you need to be familiar with. The EF, which is the edema factor, the PA, which is the protective antigen, and the LF, which is the lethal factor. So those 3 parts make up that anthrax toxin. So usually anthrax is a livestock disease worldwide. Like I said, we contract it from contaminated animals and animal products. Anthrax is also known as the rag picker's disease or the wool sorter's disease. So this is another term that you can relate to anthrax. There are 3 forms of anthrax disease in humans. There's the cutaneous. This form, you end up with a wound that forms something called a black eschar, which is like a scab. The second form of anthrax is inhalation anthrax, quite severe. And the third is the gastrointestinal anthrax, which has never been reported in the U.S. So the GI we haven't seen here in the U.S. It is treatable with penicillin and ciprofloxacin. So once identified, it is treatable. So the second pathogen I want to talk about is Bacillus cereus. This causes food poisoning, or known as a food poisoning. There are 2 forms. There's a diarrheal form. This is associated with meats and poultries, and then there is an emetic form. Emetic is, primarily vomiting, and this is associated with fried rice. So there's those 2 forms of food poisoning caused by Bacillus cereus. However, the difference between Bacillus anthracis and Bacillus cereus on a blood agar plate is that Bacillus cereus is beta hemolytic, and it is also quite resistant to penicillin. So know that those are some differences, and that can help you to identify your organism in the laboratory. So that concludes Chapter 16, Aerobic Gram-Positive Bacilli. Quite a bit of information, very important stuff. So let's move on to Chapter 17. Chapter 17 was all about gram-negative Neisseria species and Moraxella catarrhalis in particular. So with this chapter, I'm going to remind you that you need to know carbohydrate reactions for the Neisseria species that are mentioned in your PowerPoints and for Moraxella catarrhalis, which is asaccharidic. So remember that. So let's talk about Neisseria first. Neisseria are gram-negative diplococci. So they look like little kidney beans squished together, or a little baby bottom, is what I like to think of. They are unique in that they're capnophilic, which means they like increased CO2. They're non-motile. There are two primary pathogens of Neisseria to be concerned with. The first one is Neisseria gonorrhoeae, and the second one is Neisseria meningitidis. So let's talk about Neisseria gonorrhoeae first. Neisseria gonorrhoeae is humans are the only natural host for this particular organism. There are 5 colony types, and this is based on the presence or absence of pili. They're labeled as T1 through T5, and they can be grouped into 2 groups. The first group includes T1 and T2, and these are virulent. These are positive for pili. And then the second group is T3 through T5, and these are non-virulent types because they lack pili. They're pili negative. Neisseria gonorrhoeae has a capsule. It also has LPS, or lipopolysaccharide, that helps to prevent phagocytosis and facilitate infection. Neisseria gonorrhoeae is an STD known as gonorrhea. 95% of males are symptomatic, and diagnostically, if there are intracellular gram-negative diplococci in urethral drainage from a male, it is diagnostic as that patient having gonorrhea. Also, there are 3 to 5% of males that are asymptomatic, and these asymptomatic males carry a strain called an AHU strain. This AHU strain is, well, that's just it. We'll talk about it again here in a minute. So know that 3 to 5% of non or asymptomatic males have this AHU strain of gonorrhea. So for females, 50% of females can be asymptomatic, which isn't really good because treatment is necessary. Untreated gonorrhea can cause pelvic inflammatory disease and another thing called Fitzhugh-Curtis syndrome, which is a perihepatitis if it's not treated. Also, Neisseria gonorrhoeae can cause ophthalmia neonatorum, which is an infection of the eye that's acquired at birth by babies as they pass through the birthing canal. However, this is treatable with silver nitrate. If left untreated, a baby can become blind. So oftentimes this fastidious organism has to be collected and transported in a very controlled environment because it is so fastidious. They oftentimes use something called a Gembeck system, which we've talked about. It has the correct environment for it, and it's collected at the bedside. We culture it with MTM agar, or modified Thayer-Martin agar, which contains, remember the acronym TV-CAN, trimethoprim, vancomycin, colistin, and nystatin. you will see small tan colonies on your MTM plate, and know that this is glucose positive on a carbohydrate panel only. So it is only glucose positive. All isolates are tested for the production of beta-lactamase because the primary treatment for gonorrhea is cephalosporin, which is a beta-lactam. So that is why these isolates are tested. Let's talk about the other organism, Neisseria meningitidis, or the second organism, Neisseria meningitidis. This is the etiologic agent of endemic and epidemic meningitis. Up to 30% of the population are natural asymptomatic carriers in the oropharyngeal area of this organism. There are 9 serotypes based on capsular polysaccharides. Most prevalent of these serotypes is A, B, C, Y, and a serotype called W135. So serotype A is associated with pandemics. B and C are most common in the US. Y is the primary causative agent of meningococcal pneumonia in dorms and barracks. And then the serogroup W135 is responsible for invasive disease. So that is a really, really bad one. This particular organism can cause DIC. It can cause Waterhouse-Friderichsen syndrome, which is hemorrhage in the adrenal glands. Sorry. Okay, continue on. This organism grows on blood agar plate and a chocolate agar plate. Colonies appear small and gray. Neisseria meningitidis is treated with penicillin. Know that it is glucose and maltose positive on a carbohydrate panel. So it is only glucose and maltose positive. There are vaccines available for serotypes A, C, Y, and W135, but there is no vaccine for serotype B. And this is because humans, for some reason, do not develop antibodies to serotype B. There are some non-pathogenic Neisseria species, and this includes Neisseria cinerea, Neisseria lactamica, and Neisseria ceca. These are normal flora in the lower respiratory tract. So know that there are some other Neisseria that are non-pathogenic, and know their carbohydrate reactions as well. So the last organism we're going to talk about for this chapter is Moraxella catarrhalis. And this is an upper respiratory tract commensal. However, it is an opportunist, and it is the third most common cause of sinusitis and otitis media, or middle ear infections in children. 98% of isolates produce beta-lactamase, which means that you cannot treat this organism infection with penicillin. Colonies on the blood agar plate or chocolate agar plate have this this little known trait that gives them the name of a hockey puck or a wagon wheel, and it's because the entire colony can scoot across the agar. It's kind of neat to perform. And just know that Moraxella catarrhalis is asaccharidic on your carbohydrate panel, so it does not metabolize any sugars. So that summarizes 17, so Neisseria and Moraxella. So let's continue on, chapter 18. Chapter 18 is discussing Haemophilus and other fastidious gram-negative rods. Haemophilus and these other fastidious gram-negative bacilli require, they have special growth requirements, especially Haemophilus. They can only grow on chocolate agar plates because, like we've discussed, the red blood cells are lysed in that process of making a chocolate agar plate, and the red cell lysis releases factor 10, which is hemin, and factor 5, which is NAD. So anyway, Haemophilus requires these preformed growth factors. Most all pathogenic Haemophilus require both X and V, except species with the prefix para, like parainfluenzae, they only require factor 5. So know that. Haemophilus literally means blood lover. These are gram-negative pleomorphic rods, coccobacilli. They are aerobic or facultative anaerobic. They're oxidase and catalase positive. Haemophilus is also an obligate parasite. There are 10 species of Haemophilus. Only 3 are major pathogens, and this includes Haemophilus influenzae, Haemophilus aegyptius, and Haemophilus ducreyi. So let's talk about Haemophilus influenzae. This was erroneously named, if you remember, during influenza pandemic of 1889-1890. It was actually a secondary organism found in the respiratory tracts of patients that were infected with influenza or had died from influenza. They have a capsule, which is their most important virulence factor. There are 6 serotypes, A through F. The most invasive infections are caused by serotype B, so it's also denoted as HIB, Haemophilus influenza B. Not all strains are encapsulated. So because of this, there are 2 disease patterns that have emerged. There's an invasive disease, and these are caused by encapsulated strains, and then there is the non-encapsulated strains causing a localized disease. Clinical manifestations caused by Haemophilus influenzae include meningitis, arthritis, cellulitis, pneumonia, sinusitis, and in particular, it is the primary cause of epiglottitis in pediatric patients. So know that Haemophilus influenzae causes epiglottitis in pediatric patients. Another organism of Haemophilus is Haemophilus aegyptius, and this is the causative agent of pink eye, contagious conjunctivitis. Pink eye, or this organism, Haemophilus aegyptius, is also known as the Koch-Weeks bacillus or Koch-Weeks bacillus. There is another organism called Haemophilus influenzae biogroup aegyptius, and this organism produces or causes Brazilian purpuric fever. There's another organism called Haemophilus ducreyi, and this causes chancroid, which is an STD. It is also known as genital ulcerative disease, or GUD. This particular disease includes lymph node buboes, which are draining inguinal lymph nodes that develop. We have to collect and transport these quickly. They grow on chocolate agar plates. They require increased CO2, so they're capnophilic, and then oftentimes we use the Gembeck system to collect and transport these organisms. Especially Haemophilus ducreyi, it really needs to be processed within 10 minutes and viewed under the microscope in order to recover this organism. On a gram stain, you'll see something called a school of fish sometimes. So it's this formation that looks like a school of fish, where it's just a collection of these organisms. So it's just a morphology known related to Haemophilus ducreyi. Also, with Haemophilus species, you'll see growth on a chocolate agar plate, but you will not see growth on blood agar plate. So this is very important when in the laboratory. If you ever see this presentation where most all organisms will grow on that multi-purpose general blood agar plate. However, with Haemophilus, because they require those preformed factors, you'll only see growth on your chocolate agar plate and not on that blood. So right away you should think to yourself, this is a Haemophilus species possibly. Let's move on to some other fastidious gram-negative rods. One of these groups is called the Hayseck group, and these are quite fastidious organisms. They include 5 different species, well, 4 species and then 1 group of species. They're all important causes of endocarditis. They are normal flora of the mouth, and they are opportunistic organisms. So the first one is Haemophilus ephrophilus. Next one is Actinobacillus actinomycetum comitans. This organism is connected to subacute bacterial endocarditis and have a star-shaped center in their colonies. The next organism species is Cardiobacterium hominis, and this is, you'll see a unique rosette formation on the gram stain. The next organism is Eikenella corrodens, and this is implicated with bites and fighting, human fights. This causes cellulitis, and this particular organism is well known for pitting the agar, and it has a bleach-like odor. And then the last group of species that's in this Hayseck group is the Kiniella species. So there are 5 particular organisms that you need to know in this group, and a few of those important details that I've just stated. Another group of fastidious gram-negative organisms is a group called the Capnophaga. These are gram-negative fusiform rods. They are also a normal mouth flora. A unique characteristic about Capnocytophaga species is that they have a gliding motility property. Another fastidious group of gram-negatives is the Bordetella species. These are gram-negative coccobacilli. There are 7 species, however, 2 are primary pathogens. These are obligate aerobes. So there are these 2 primary pathogens of Bordetella are Bordetella pertussis and Bordetella parapertussis. Their ability to cause pathogenicity is related to their ability to adhere to and replicate on ciliated epithelial cells. So the first organism, pathogen, Bordetella pertussis, causes the disease known as pertussis, and this is also called whooping cough. There are 3 phases to this disease. The first phase is catarrhal, or catarrhal. The second phase is paroxysmal, and in this second phase is where you get that characteristic whooping cough. And then the third phase is the convalescent phase. I just want you to know that there are two primary augers we use to isolate and differentiate, select and differentiate these organisms. The first one is the Bordeaux-Genjou auger, and this particular organisms will appear as half pearls, half pearl colonies on the Bordeaux-Genjou auger. And then there's the Regan-Low auger that the colonies will appear as mercury droplets. Pertussis is treatable with erythromycin. So just know those two pathogens. The next group of fastidious gram-negatives is the Brucella species, or Brucella group. This group is a CDC bioterroristic agent. It is gram-negative pleomorphic rod. It is a zoonotic infection. It causes the disease brucellosis, also known as undulant fever. There's a stage of this disease where you have this undulating fever that comes and goes. Infection to humans is caused by contamination with milk and or animal exposure through occupation. The best culture source is bone marrow and blood, so know that for Brucella. There are only 6 species that are pathogenic to man. So know there's only 6 species pathogenic, but many more in this group. The most common cause of brucellosis is the particular species Brucella melitensis. So know that melitensis is the most common cause of brucellosis. There are 3 phases to brucellosis. This is the acute, subchronic, and chronic. Be familiar with that. The next group of fastidious gram-negative organisms is Francisella. This is also a CDC bioterrorism agent. These are gram-negative pleomorphic rods as well. There is only one particular species that is implicated in disease, and this is Francisella tularensis. This causes the disease tularemia, also known as rabbit fever. It's contracted through exposure to animals or tick bites. The next fastidious gram-negative organism is Legionella. These are gram-negative pleomorphic rods, ubiquitous in nature. There are 50 known species. 20 have been isolated from humans. The primary pathogen in this group of organisms is Legionella pneumophila. There's 2 forms to the disease that it causes. The first disease is called Legionnaire's disease. The second one is, and Legionnaire's disease is a pneumonic form, and it's the most common manifestation of Legionella pneumophila. But there is another non-pneumonic form of Legionella, and this is called Pontiac fever. So it's spread through the blood. So this one is a non-pneumonia secondary to Legionnaire's disease. Legionella pneumophila is asaccharitic, so know that. It resists water treatment, making it a common major reservoir. The best way to detect this particular disease is in urine. There's a urine antigen test that is used to detect it. It's very fastidious, only growing on chocolate agar plate. It's a slow grower and also requires L-cysteine for isolation in the lab. We can grow it on buffered charcoal extract or buffered charcoal yeast extract agar, BCYE. Colonies appear as ground glass, and they are also fluorescent under long wave UV light. So 2 unique things there. It's treated with ciprofloxacin. So there's a treatment. And the last group of organisms that we'll talk about that are fastidious gram-negatives are Pasteurella. Pasteurella species are gram-negative pleomorphic rods. However, these are bipolar staining. Note that they grow on blood and chocolate, but uniquely, they do not grow on EMB or MacConkey plates. So this kind of makes it difficult for detecting them right off the bat because they're gram-negative, which EMB and MacConkey select for gram-negative. So this is a gram-negative that uniquely does not grow on those selective plates. Usually this particular infection or with one of these organisms is acquired from animal bites, a dog or a cat. And usually this leads to cellulitis, and the causative species in most all of these cases is by an organism called Pasteurella multocida. So that's all that I think you need to know for Chapter 18. So let's go on to the last chapter in this exam review, which is Chapter 19. And that was a big one. This one includes the entire huge family of Enterobacteriaceae, also known as the enterics. It's a huge group, a huge group. They all look alike. They're facultative anaerobes. They're gram-negative rods. They all ferment glucose. They're all motile at body temperature except for Klebsiella, Shigella, and Yersinia. They're all nitrate positive and oxidase negative. The definitive ID of these organisms depends on biochemical reactions, carbohydrate reactions and biochemical reactions. So there's a lot of testing involved when you're identifying these enterics. There are a few very key characteristics, though, in the micro lab that will lead you in one direction or the other. So let's just jump right in. So there are, in this family of enterics, there are 3 cellular antigens that are common to most all these organisms. These antigens are the O, the H, and the K. The O is the somatic antigen. This one is heat stable. And then the H antigen is the flagellar antigen. And then the K antigen is the capsular antigen. So the H and the K antigens are heat labile, meaning they're destroyed by heat. So if you broke this family into 2 categories, you have opportunistic pathogens and then you have primary pathogens. And your primary pathogens will cause disease in any human, adult, person, young, old. They're a true pathogen, so they will cause disease in anybody, healthy or not. Your opportunistic pathogens are usually cause infections in immunocompromised patients, or they'll be nosocomial, caused by hospitalization, or maybe a procedure that was performed like catheterization, or given an ability to get into your body where it doesn't belong. So let's just jump right in. Well, one note before I continue. Of your primary true pathogens, there are only 3 that we'll discuss. All the rest are opportunistic. And those 3 primary pathogens are Salmonella, Shigella, and Yersinia. So let's talk about our opportunistic pathogens in the Enterobacteriaceae family. The first big group is Escherichia coli, or E. coli. These appear with a green, beautiful, brilliant sheen on EMB agar. There are 7 subgroups. They all have O, H, and K antigens, And if you remember about the IMVIC reactions, the indole, methyl red, Voges, Proskauer, and citrate reactions, this one is a positive, positive, negative, negative for IMVIC. E. coli ferments glucose, lactose, trehalose, and xylose. And those 7 subgroups include 1, uropathogenic E. coli, and this is your primary cause of urinary tract infections. You have second subgroup, which is enterotoxigenic, and this is known as traveler's diarrhea. Your third subgroup is enteroinvasive. This one primarily causes dysentery. Your fourth subgroup is enteropathogenic, which is infantile diarrhea. And then your fifth subgroup is enterohemorrhagic, and this is your O157H7 strain. This one leads to hemolytic uremic syndrome. Also has 2 cytotoxins. They're called virotoxin 1 and virotoxin 2. And this particular subgroup is sorbitol negative. So this sorbitol negative makes it unique. We screen for this with a SMAC plate, a sorbitol McConkey plate, and these organisms will appear colorless. They do not ferment sorbitol, whereas all your other E. coli's will ferment sorbitol. Your 6th subgroup is the enteroadherent E. coli, and your 7th subgroup is the extraintestinal E. coli infections, usually isolated in a blood culture as a contamination. So the next group, so our first group was E. coli. Our second group is a group that includes Klebsiella, Enterobacter, Serratia, Pantoa, and Hafnia species. Klebsiella, Klebsiella in particular, has that green sheen as well on an EMB agar plate. So when you initially set up a stool for culture and you see that green sheen maybe on an EMB, you can't right away just say that's E. coli. It could be Klebsiella. But when you do biochemical testing, you'll find that Klebsiella in its IMVIC reaction is positive, negative, positive, positive. The others in this group right here are IMVIC negative, negative, positive, positive. So they're the opposite. They're the opposite reactions to E. coli. Let's talk more specifically about Klebsiella. This is also known as Friedlander's bacillus, and it causes pneumonia. Klebsiella pneumoniae is identical to Klebsiella oxytosa, except oxytosa is indole positive and Klebsiella pneumoniae is indole negative. Let's talk about Enterobacter and Pantoa. These can be differentiated from Klebsiella because they're very similar to Klebsiella, but they can be differentiated from Klebsiella based on their ornithine decarboxylase test. Klebsiella is ODC negative, whereas Enterobacter species are ODC positive. Let's talk about Serratia. Serratia, there are 3 primary species that you may come across in the lab, and that's Serratia marcescens, Serratia plumuthica, and Rubidea. These produce a red-orange pigmented colony. So know that Serratia species can produce a red pigmented colony. Group number 3 of are opportunistic Enterobacteriaceae, includes Proteus, Morganella, and Providencia species. All of these are non-lactose fermenters. They're normal intestinal flora. However, Proteus is clinically significant. There are three particular organisms, Proteus mirabilis, Proteus vulgaris, and Proteus pinneri, that are clinically significant. And they display this swarming growth on plates. So they'll swarm the entire plate, take over the entire surface. They also have a burnt chocolate smell, a unique burnt chocolate odor. So Proteus mirabilis and vulgaris are very similar species. So two tests that will help you differentiate between those two particular species are indole and your ornithine decarboxylase test. P. mirabilis will be indole negative, whereas P. vulgaris will be indole positive. Mirabilis will be ODC positive, and vulgaris will be ODC negative. Also, this group of organisms is PAD positive. So these will differentiate. This test, PAD test, will differentiate from the rest of the Enterobacteriaceae family. So it's another unique test that will help you differentiate this group from the rest. Morganella is a non-swarmer. There is one particular organism, Morganella morganii, that causes urinary tract infections. And then Providencia, there are several clinically significant, but they're not a common organism we see in the lab. So there are several clinically significant Providencia species, but that's all you really need to know for now. The fourth group of Enterobacteriaceae opportunistic organisms is Edwardsiella. This particular group of organisms is implicated in bacteremias and wound infections. And then your fifth group is Edwinia pectobacterium, and this is primarily a plant pathogen. Your group 6, or sixth group of opportunistic Enterobacteriaceae, is the Citrobacter family, or group. And these are very similar to E. coli. We differentiate primarily with the citrate reaction. Another note, Citrobacter frundii, this particular organism is similar to Salmonella, and we would differentiate with urease and lysine decarboxylase, or LDC test. Citrobacter frundii is urease positive, LDC negative, whereas Salmonella is urease negative, LDC positive. So to differentiate between those two very similar looking organisms, we would do those two tests. Your seventh group in this family of Enterobacteriaceae is your primary pathogens. So these are primary pathogens of the intestine, including Salmonella species, Shigella species, and Yersinia species. They're all non-lactose fermenters, and they're H2S positive. So this makes them unique. Most of your other enterics are lactose fermenters. But these three primary pathogens are non-lactose fermenters. Salmonella, the first primary pathogen, has two main species to be concerned with, and this is Salmonella enterica and Salmonella bongori. Let's see, it's Imbach reaction. Know that it's indole negative and Voges-Proskauer negative. It has O and H antigens, which is used to determine its serotype. and its VI antigen, it has something called a VI antigen, primarily identifies the serotype typhi, so Salmonella typhi. So that particular serotype, Salmonella typhi, causes something called typhoid fever, which is a gastroenteritis from contaminated food. People can harbor this organism in their gallbladder for long periods of time, or indefinitely, actually. And this is considered a carrier state. So Salmonella typhi can be harbored in your gallbladder. The next primary pathogen group, Shigella, is also a non-lactose fermenter, and it is H2S negative. So differentiating between Salmonella and Shigella, you can do the H2S. You can use H2S. Salmonella is positive, Shigella is negative. Shigella is the primary cause of bacterial dysentery. There are 4 major species based on the O antigen serotype. There's Shigella dysentery, Shigella flexneri, Shigella boydii, and Shigella soni. So dysentery is group A, flexneri is group B, boydii is group C, and soni is group D. Soni, Shigella soni, is the first most common in the U.S. and that second to soni is Shigella flexneri, so know that. Infection can cause serious complications, causing ileus, which can lead to something called toxic megacolon. So just remember that info. There's also another important point. In your PowerPoints, there was a slide that mentioned serologic grouping, and it just states briefly about if agglutination fails with Shigella antisera, the next thing you would do would be to heat the suspension to 100 degrees for 10 minutes. The reason you do this is to remove capsular antigen VI. So after that's removed, then they can repeat with that antisera to determine serologically what serotype you have. So it's a testing method to differentiate serotypes in the laboratory. So the next true pathogen is Yersinia. This is also a non-lactose fermenter. This one is a short plump rod, gram-negative short plump rod, and it appears as a safety pin. Unique about this organism is that it grows 25 to 30 degrees Celsius. So this is another organism that can be cold enriched, or cold enrichment can be used for recovery. It grows better at slightly colder than body temperatures. So Yersinia causes plague in rodents, and it is transmitted to humans via a rat flea. There are 2 forms of disease. There is the bubonic plague, which is a glandular plague or glandular form. And then the second form is a pneumonic plague, and this is secondary to the bubonic infection. And this is spread systemically through the blood. 2 particular species are Yersinia pestis and Yersinia enterocolitica. Enterocolitica is the most commonly isolated species. in clinical infections. So know that. We can grow this organism on something called CIN agar, which includes cephalodin, irigasin, and novabiocin. And this has a unique red-centered bullseye in its colony. So the colonies will have these red-centered bullseye appearance. There's one more organism called Yersinia pseudotuberculosis, and this is primarily found in guinea pigs. It's a pathogen to guinea pigs and causes these caseous swellings called pseudotubercles. It's most of the time fatal in animals and rare infections in humans. It's rarely found in human infections. So that's Yersinia. So we got pestis, enterocolitica, and pseudotuberculosis. And I believe that is a wrap for chapter 19, that great big family of Enterobacteriaceae. I think that covers everything, and if you followed along and just made notes or highlighted in your PowerPoints the things that I mentioned, you'll ace your exam. So that was a big review. I know it was, but it's been, like I said, quite a while since we discussed this and learned it. So anyway, that is that. Exam 3 review. See you soon.